Anyways, thanks for your introduction. I think I'm the second last presenter. Please hold on for another 40 minutes, then we can enjoy our dinner. So I would like to talk about something about characterization of root senescence in Barley. Normally, when we start our topic, the first thing is we emphasize how important our work could be. So why study root senescence? Because root senescence is important for the global carbon cycling. For example, here, plant fix carbon via photosynthesis, and then carbon is released to the soil via carbohydrates or root senescence. In another word, root senescence is an important part of global carbon cycling. Second, in agronomy, root senescence is also good if we better understand it, because understand root longevity could help to increase fertilizer while reduce fertilizer input. For example, from these slides, at the last stage of green filling, there is a huge demand of nutrients and water. This is also a critical stage for root senescence. So there is a conflict between green filling and root senescence. If, for example, we could better understand the senescence of cluster roots, probably Professor Lambert would be very happy to know about it because it will increase the P-use efficiency. But even this terminology has used for several times, the definition is still very widely defined. For example, aging versus senescence. Here, in my study, I always consider aging as a time-dependent biological process at any developmental time and any developmental phase, meaning it's only time-dependent, time-relevant, not biological relevant. While I define senescence in this way, also from literatures. It describes a genetically controlled developmental program with the purpose of nutrients remobilization at organ or whole plant level. And it is not well described in roots. Actually, from literature, so far as we know, especially from ecology, root browning is widely used to distinguish young and old roots. While we look at anatomy of roots from many publications, cortical senescence is an indication of root senescence, but how these two parameters temporally and spatially correlated remains unexplained. Therefore, we ask, are roots also senescing? I mean, is it similar as what we know in leaf? For example, in leaf, we know degradation of chlorophyll, but there is no chlorophyll in roots. Therefore, what is the biological process in root senescence? To answer this question, we set up our aims of the goal. We want to describe aging process in barley roots. Second, we want to verify whether this process reflects senescence. To answer these two questions, we set up our experiment in a hydroponic condition, and we grew them for 2 months for barley. So here is the phenotype, what we observed. Day 18 means 18 days after germination. The left part is always seminal root, which is embryonic root. The right part is always nodal root, which is post-embryonic root. So later on, when I talk about senescence, I always only talk about seminal root senescence because there are two different root types. Now, let's quantify this picture, shall we? So first of all, let's look at the above ground. So we quantify the chlorophyll concentration of individual leaves of the main tiller. At day 18, there are 4 leaves of the main tiller. This is the distribution of chlorophyll concentration. 1 week later, 2 more leaves, and first leaf is already senesced. So with this picture, we conclude there is a progressing of leaf senescence in the above ground in barley, in the main tiller. How about in the below ground? We first look at the seminal root. Of course, it's the first emerged root system. For seminal root, the biomass of seminal root was arrested at day 46, while its total seminal root length was arrested 1 week earlier. With this picture, we conclude we might capture a senescence process in barley seminal root. Let's look at the nodal root system. Nodal root biomass is just happily increased. Therefore, there might be no senescence in nodal root in our window. So later on, we only put our focus on seminal roots. Now, look at anatomy, one step deeper. We have 3 sections at 3 levels, 5 centimeters under the hypocortical. We also observe the phenotype of the root tip, seminal root tip. There is more or less no significant change. Also, we stain them with Evans blue, which is a dye for dying or dead cell. But from day 39 on, we observed cortical senescence, not from somewhere else, but from the old part. And then, during the progressing of time, cortical senescence spread from old tissue to young tissue. From the tip, we also observe a degradation of root tips. This is a new phenotype we just observed. So now, we are at the anatomical level. We then spread our study into physiological level. How about activity? For leaf senescence, we know leaf senescence coincides with the decrease of leaf activity. For example, photosynthesis activity. How about root? We use N15 uptake capacity as an indicator of root activity. So the red line, nitrate uptake capacity was almost maintained at a seminal level before day 39 and then dropped significantly. And there is a slight increase of translocation of nitrogen, but also sharply decreased. Therefore, root activity sharply decreased after day 39. And this is actually a little bit different compared with literatures, because some literatures, by dissemination study, they assume nitrate uptake capacity just continuously decreased during root aging. In my study with hard data, we conclude maybe for root systems, they have a capacity to maintain the uptake rate well before the decrease when senescence set in. When we look at the sugars, why look at this? Because normally scientists speculate the decline of root activity is due to depletion of energy. This is logic. But actually, at day 39, before or after, we didn't find a significant decrease of sugars. Therefore, it looks like photoassimilase is not an influential factor for the decrease of root activity. We also look at spike development at day 39. Actually, the spike of the main tail is just about 1 millimeter long or 2 millimeter long. Actually, it's a very, very small thing. It is not likely that the spike can overcome the assimilate demand compared with roots. Therefore, these two together, we conclude maybe root senescence is a result of endogenous regulatory network rather than a result of the shoot-dependent regulation. We go one step further, again, at physiological level. During leaf senescence, normally scientists get a decrease of soluble proteins. Actually, we didn't get it during root aging process, but we do get a decrease of total amino acids. When we look at these amino acids individually, we can see that at a global level, actually, more or less, they change at day 39, either decrease or increase. For example, here, phenylalanine. Phenylalanine is a precursor of salicylic acid. Actually, we also get accumulation of salicylic acid during root aging. Specifically, I'm interested into tryptophan because in some rice papers, tryptophan has been identified as leaf senescence markers. Tryptophan is a precursor of tryptamine. Tryptamine is a precursor of serotonin. They both should gradually increasing abundance during root aging. Therefore, the same markers in leaf senescence could serve similar functions maybe in root aging as well. With this, we go to very fundamental questions. What is the purpose of senescence? In leaf, we know for leaf senescence, the purpose is nutrient remobilization. Do we also find the similar result in root? We are plant nutrition group. We analyze all possible nutrients we could, and actually, we could only identify 3 possible elements that should decrease their pool size, which is content, after day 39. Their concentration, the red bar, continuously decrease, but their content roughly only decrease after day 39. So the pool size just decrease, indicating maybe they are remobilized to somewhere else. So our data indicates probably, but need more verification, probably nutrient remobilization also happened during root senescence, but not that many. Mostly, I think you are interested into nitrogen, yeah? During fine root senescence, at least we know nitrogen concentration declined, but in our system, concentration of nitrogen rather stand at a similar level. Its content is more or less like a biomass curve. Therefore, the pool size of nitrogen didn't decrease during root aging, indicating probably nitrogen was not remobilized during root aging. We also did a transcriptome analysis, but in this we play a little bit. We defined a root tip, which is from root cap to first 1 mm root, and the rest of seminal roots. Why we do this? Because in senescence studies, there is an argument which factor dominates the aging process, either tissue age or plant age. For example, a branch from 100-year spruce tree, their rooting percentage will be extremely low because the plant age dominates the rooting percentage. But if you use a very young tree, the branch rooting will be very high. So therefore, aging, especially plant aging, play a role. We use tip because root tip is continuously elongating. More or less, their change only reflect plant age because tissue age is very similar. But the basal part reflects both plant age and tissue age. By transcriptome analysis, we use day 18 as a reference, then calculate the gene fold change for later developmental stages. We first did a non-supervised clustering. Actually, we get extremely similar clustering between these two different segments. For example, gradually down or upregulated, I'm not surprised because plant aging is gradually upregulated. Interestingly, at day 32, which is 1 week before the phenotype, we get huge amount of upregulating genes. And actually, this cluster contains huge amount of transcription factors. They are probably involved in the regulation of root senescence. We then do a GO term analysis at day 25. There is no GO term could be enriched. But for the significant gene at apical zone and basal zone at day 32, in the middle part, which share almost 50% of the genes, actually, we enrich the GO terms. They are dominated by regulatory genes. For example, here would be protein phosphorylation and regulation of transcription. And they maintained at this time point. Somehow, after day 39, the dominant GO term changed, switched to an oxidation reduction process. And this gets more important according to the number and p-value, indicating day 18 at transcriptional level, it's a changing phase for root senescence. I'm pretty interested into those transcription regulation GO terms. In this, it contains huge amount of transcription factors, and these are dominated by NAC, WRKI, and AP2 transcription factors. They show very sharp upregulation at day 32. In this, we identified 2 transcription factors. They are pretty interesting because they showed root aging specific upregulation, but not leaf aging upregulation. Probably indicate these 2 have a specific role only in root senescence, but not in leaf senescence. We quantify our results by several analyzed gene markers. They show similar results as we expected. For example, here, PAP17 is SAC12. It's a golden marker for leaf senescence. It showed upregulation as well. We analyze hormone profiles. I'm not surprised by the decline of auxin, decline of cytokinin. It's expected. Surprisingly, we get a very sharp peak of ABA at day 39. Just coincides with the phenotype we have at that time point. And it is confirmed by ABA biosynthesized genes, NCED1 and NCED2. Therefore, we go back to our definition. Senescence is a genetically controlled developmental program with the purpose of nutrient remobilization. Our transcriptome data indicates a chronological sequence of regulatory events support this part, and also we have other evidence support the second part. We identified the possible remobilization of nutrients. Of course, we need more data to support this. With our data, we conclude, yes, roots undergo senescence, and ABA might be a trigger for root senescence. With the knowledge we have, we extend our research to compare different root types. For seminal and nodal roots, each circle indicates a biological replication. We have 5. And then for each circle, we do proteomic profiling. And with proteomic profiling, we actually can distinguish seminal and nodal roots nicely by PCA. Their dynamics also show very similar clusters also. Even nodal root emerge later than seminal roots. And by clustering, by goal term analysis, we pick one goal term, which is hydrogen peroxide. And in that, we identified a protein, which is just nicely showed accumulation during aging. Probably in the future, this protein could be a marker for root senescence. With this, we would like to thank my supervisor, Nico from UVN, and cooperation with Professor Sebastian Carpentier. I also get a huge amount of help from my colleagues to study this long-term and large-scale hydroponics. And also, I'm looking for a postdoc position. I would be happy to talk about the possibilities. Thank you very much.